Good afternoon, everyone. It's 3:03 p.m. So let's get started. Today, our speaker is Dr. Vincent Michael. Vincent is currently a postdoc on the tropical food and breeding genetics program. He did his bachelor and master's back in India in plant breeding and genetics. After he got his master's, he worked at the IPRESET, which is a major national institute work on sorghum, millet, and as well as legumes. Vincent joined TREC in 2017 as a PhD student in the Western Breeding Genetics Lab under Dr. Jeffrey Monroe. He was working on crown road on root squash. Graduated in 2021 and become a postdoc in the tropical fruit and genetics breeding program, work on tropical fruits, including avocados and mangoes. Vincent is very experienced and trained plant breeder by training in both conventional and modern breeding techniques. He's very skilled at quantitative genetics, and he's spearheading into this new genomic area where to use all kinds of tools to that can be used for to speed up the breeding cycles, especially for tropical and ornamental crops that are really facing long breeding cycles and hard to have a new cultivar release. So without further ado, Vincent, the floor is yours.
Thank you, Shingbo, for the introduction. So today I'm going to talk about project that we did on mango seedling genotyping, which from the results we got revealed some potential self-incompatibility in some mango cultivars and also some pollinator behavior from the group that we have in TREC. So mango belongs to the Mangifera indica species, and this species is in the Anacardiaceae family, which also has important crops such as cashew, pistachio, poison ivy, and marula, famous for the marula drink. And it's in the genus Mangifera. Now, within this genus, there are other species that also produce edible fruit, similar to mango. So the first image on the left is the mango, which is Mangifera indica. And then we have the other, some other wild species, such as the Lanica, Laurina, and Kasturi, which produce edible fruit. So this fruit was domesticated in India about 2,000 years BC. And from there, migration patterns first went to the southeast, into the Pacific Islands, and then to Africa, then to Latin America and South America, and in the mid-1800s to Florida. The same time that it was introduced in Florida, it was also introduced in Australia, more or less around the same time. Now, the cultivation is only within the tropical warm climate. So as you saw in the migration patterns, this is just the, the areas where it was introduced is where it could do best. And it's only restricted in those in that region. So most of the cultivation is in India and China, followed by Mexico also has a huge cultivation acreage, and South America, and a number of countries in Africa. India is by far the largest producer of mangoes. More than half of the, it's a lot of it. And then followed by China, Indonesia. All these are mostly the countries in Asia. And then in South America, we have Mexico and Brazil, which have high production. And the only African country is Malawi, and then the rest of the world share the about 15.8 tons. Now, for this talk, I want to concentrate on breeding. So I'm gonna just give you an overview of the breeding strategies that have been used in mango over the years. The first one is mutation breeding, where people use gamma irradiation to induce mutation or chemical agents. The other one is controlled close pollination, where you manually hand pollinate the mango flower. Sometimes you have to do emasculation to remove the pollen, and then bag the plant when you've done the pollination to ensure that no contamination occurs from pollen from other plants. And then there's controlled open pollination, which is you have your plants growing out in the field, and then you put a cage over them to just have only those within the cage pollinating and no pollen from outside. So the cage is just to prevent pollen from other surrounding trees getting into the cage and contaminate maybe the plant crossing. And the final one is seedling selection, which just means that you let the natural outcrossing take place, then you collect the seedlings at the fruits and get the seeds and plant them as seedlings and select the ones that have superior qualities. So of course there have been, varied measures of success within these methods. So mutation breeding basically is not used anymore, and there have been very little success with it. The manual pollination is, most breeders use it, but you always end up with low success rates, very few hybrids. And then there is issues of incompatibility between the two parents that maybe you want to pollinate, and flower synchronization, that the flowering times may be different. So there's that limitation. Now for the controlled open pollination that happens in a cage, the pollination works successfully, but then you will have to evaluate a lot of seedlings, basically all the fruit that you get from within that cage, to select your superior hybrids. And then you will not be able to identify any cells from those seedlings until they mature and produce a fruit. Then you can compare that to the parents that you used, and you can tell, okay, this was a self and not a cross. And the trees that you're using for the cage have to be in close proximity. If they are far apart, it's very difficult to, house them within a cage. Now, seedling selection, believe it or not, has had the most success, but it's also limited because you have to evaluate all the seedlings you obtain, and you can't also identify self-pollination. But you will not also know who the paternal parent is when you select seedlings by, that have naturally outcrossed. But it has been very, very successful. And in Florida, especially, there have been a number of cultivars, the major cultivars that we have here, that were selected as seedlings and became very profitable and widely adopted for commercial production, including Hayden, Arwin, Tommy Atkins. Now, as you can see from this table, so we have the cultivar on the first column, and then on the last two columns is the date when the seedling was planted and when the first fruit was harvested. So looking at those dates, it's clear that this takes a long time, minimum of 5-6 years before you can get the first fruit. So this is one of the other major limitations for seedling selection in this manner, but it has been successful. So remember this image I showed earlier about the migration? Because of the success of seedling selection here in Florida, now most of those seedlings were adopted in many other regions across the world for commercial production. And they are very well adapted and and very valuable to the growers. Now, my question, or the question I wanted to answer is, can I take advantage of current breeding technologies to improve on the seedling selection strategy? It has been successful, but then there are some disadvantages. So my question was, can I use marker-assisted selection to address some of these challenges? So with marker-assisted selection, it's very clear that I can avoid labor-intensive pollinations techniques. It's also possible to select seedlings from desirable parents because you can identify those parents using markers. And since you can identify parents, you can also identify the self-progeny. And that means that when you take your seedlings to the field, you will only have fewer seedlings to test. You know, you already have a lot of information about the seedlings, and you have, gotten rid of the ones that are not desirable. And then it allows you to exploit the available genetic diversity. So basically, you could have a collection of diverse mango fruits and let them cross, select fruits from there and get the seedlings, and use this method to identify favorable hybrids. And with it, you can accelerate the breeding cycle for the mango. So my objectives were to use a mango single nucleotide polymorphism array to identify parentage of mango cultivars, and then test these for seedling selection from some of these cultivars. And then using that information, try to establish clues of self-incompatibility for some of them, and also maybe look at the pollinator behavior in the groups. Now, parentage of mango cultivars has been studied before, and most of those studies used, maybe I would call them second generation molecular markers. They're short sequence repeats. And usually you can only analyze few of them, and they are costly if you want to do a large analysis. Now, with the availability of the mango genome, it was possible to make single nucleotide polymorphisms for mangoes. And there was an array that was produced, and they used it to phenotype some cultivars and try to identify parentage. But you had to do this by looking visually on the SNP genotypes on an Excel sheet. And this was not feasible, it was very laborious. So because of that, there are still many open cultivated cultivars that have either a single parent or lack parentage information whatsoever. So the materials I used for my project was the cultivars that we have here in TREC. I took 140 of them, and then selected seedlings from 11 maternal parents, a total of 575 seedlings. Now, these maternal parents were monoembryonic, and they are commercially significant cultivars. So I extracted the DNA, and it was genotyped with a total of 399 markers using Fluidigm EP1 platform. And then when I got the data, the SNP data, I did the parentage analysis. So this is how I did it. When you get the data from the array, it looks like these two images. So parent one, you have the SNPs for the two DNA strands, and then parent two, you have its own SNPs. So the first thing you do, you remove the heterozygous and monomorphic SNPs. So let me point with the mouse. The monomorphic SNPs are the ones that are similar in the two parents. Basically, that means those markers are not giving you any information. You want the ones that will differentiate the parents. So we get rid of those. Then we have some others, like in this region, which are different in the two strands. So if you have an F1 from here, you're not gonna tell whether the A they have came from this parent or from this parent. So you can't really differentiate that. So you get rid of those SNPs. And after that, you have something that looks like this. So the loci in both parents are homozygous and homozygous and polymorphic, that they are different. Now with this, it's possible to predict what seedlings or offspring will come from those two parents. You basically take the first strand of the parent and pair it with the second one. And you get your genotype of the expected offspring. Now, when you have that, and you have genotyped your offspring, you can do calculation of genetic distance, which is by comparing the expected progeny and every potential offspring that you have, and then perform a Dixon test to have significant values of the parentage combination. Now, when I did this, I used a software called Apparent in R, which is a statistical software and very useful for this. It's been used for other trees, mostly peaches and apples. And then with the SNP information, I also examined the population structure using model-based Bayesian clustering, and also checked how the phylogeny of the cultivars we have in TREC differentiates. And finally, using the results of the parentage analysis, I tried to display which are the pollen donors that are amongst the cultivars that we have, and how far did the pollen travel from the donor and the recipient tree, and also check to see whether there was selfing in some progeny. And in those cases, we assume that the pollen did not travel. So these are some of the results that I got for parentage analysis of Florida cultivars. On this table, I have 5 columns. The first column is the name of the cultivar. In the second column, we have a pedigree from literature that other people had studied with SSR markers. And then parent 1 and parent 2 is the data that I got from my analysis. And the last column shows the type, which is whether it just gives you details whether it was an outcross or a cell. Now, from this table, you can clearly see that the data that I got from my SNP analysis corresponds to what had been reported before. Now, in this table, which is the columns are similar to the first one, but if you look at the pedigree from literature, there was only one parent provided. So from my data, I was able to identify that parent, the one parent, and also the second parent. Now, I've underlined Duncan because we found something interesting there. In the literature, Duncan was reported to be an offspring of Namdokmai, which is a southeast mango. But when I dug further, I saw that this cultivar was not in Florida when Duncan was patented. So there was no way it would have provided pollen for Duncan. But the patent did mention that one of the parent was Edward, which is what I got. And then from my analysis, I was able to find the second parent, which is Carabao. Now, this third table shows you a part of the results that I found where there was no information at all from literature about the parentage or from anywhere. But from my analysis, I was able to identify either one or two parents for most of these Florida cultivars. Now, when I looked at the regional cultivars, those are cultivars that are, brought from other places. It was more or less the same. So I could confirm parents for some of the ones that had information from literature, and also identify parents for the ones that didn't have any information. Then, so in summary, for the cultivar analysis, I was able to confirm 16 cultivars which had been reported before, and also identify the missing parent in all of them except these three cultivars, Brooks, Coggeshall, and Joylin. And then for a total of 67 cultivars, I identified parentage information for the first time. 33 for both parents, and for a single parent for 25 of them. And from the cultivars, I was able to identify 14 which were selves or synonymous genetically, meaning that they have different names, but they are genetically the same cultivar. But I could not identify parentage information for at least a total of 45 cultivars, including the wild accessions. Now, for the analysis of the seedlings, I have this table I'll explain to you. So the first column shows the cultivar that I used as the mother parent. These are the 11 cultivars. And then the second column is the total number of seedlings we were able to generate from the seeds we got from the mother parent. Category 1, 2, 3, and 4 correspond to the significant testing. So category 1 means it was significant at 0.05, category 2 at 0.15, category 3 at 0.2, and category 4 at 0.25. So you can see that for most of the seedlings, we were able to identify parentage information within very good significant levels. And then at the same time, in the last column, we were able to identify some selves. So for example, Tommy Atkins had the highest selfing rate. And Van Dyke, Ruby, Duncan, and Lemon Zest, they didn't have any selves. So basically what this means is for a grower, if you have Tommy Atkins in your group, only Tommy Atkins, you will get fruit. But if you have the other cultivars that don't self, you will not get any fruits. You have to introduce a different cultivar to provide pollen for fruit set. Then I also did the population structure, and this is how the population differentiates for all our cultivars here in TREC. in the top figure, it shows only 3 clusters. The one that is far right, majorly red in color, are accessions that came from Southeast Asia. And green is mostly Florida accessions and some Indian accessions, which is not surprising because most of the Florida cultivars were seedlings of Indian cultivars. And then the last section, the blue one, is Caribbean, South America, and maybe a little bit of Florida accessions as well. In the second figure, I included both the cultivars and the seedlings. And I expected to only have 2 populations. So basically the seedlings and the cultivars were separating. But in the third figure, it shows that all our seedlings were uniquely correlated with each other from seedlings from the same maternal parent, as you can see with the pattern that is showing there for each of the maternal parents. So, and then from the phylogeny tree, we got only 2 clusters, 2 major clusters. The first one containing mostly the Southeast Asian cultivars and the wild accessions. And then the second one having mostly the Florida cultivars for the first subcluster. And then Florida and some Indian cultivars for the second cluster. And the Caribbean and Latin America for some in subcluster 3 of the second cluster. Now, some of the cultivars that we have in the last one are newer cultivars. They've been released maybe in the past 15, 20 years. So they've used different materials. Now, when I included the seedlings in this figure, the pattern was as I had seen in the structure analysis. The seedlings basically clustered together with where the parents are located on the phylogeny tree. Now, what I want to bring to your attention is this cluster where Duncan is. There's maybe 1 or 2 Florida cultivars there, Joylene and one other one, and Duncan. So this cluster has a lot of potential if we want to introduce diversity in the cultivars that we have in Florida. It's a good genetic resource to tap into from this cluster. Now, with the results of the parentage, I just overlaid them on the phylogeny tree. So this one just shows the arrows, and the arrow is pointing to the maternal parent, which are highlighted in blue. And it's coming from the pollen donor. The ones that have a circle means they are selfing. They had some selfing rates, progeny detected. So you can clearly see, like Tommy Atkin, most of the pollen was just, most of the seedlings were just cells. But another thing is that almost all the seedlings that we got, the pollen came from mostly all over the group, from most of the parents. So it's a diverse collection. Then we calculated like how long the distance would be for the pollen to get from the donor to the maternal parent. And most of them were between 0 to 80 meters, although we had some pollen that traveled over 150 meters. And when we checked the maternal parents to see which which ones received pollen from the farthest tree. Palmer and Tommy Atkins had pollen from the farthest tree, but it could also be because they are located at the farthest ends of the group, far from the other maternal parents. So in summary, the parentage analysis that we used was able to identify and resolve missing pedigree information for mango cultivars in our collection. And then the collection population structure differentiated into 3 clusters, which correspond to the major geographical origins of this germplasm. The phylogeny tree only had 2 clusters, but there was some sub-clustering in the second one. And greater distance was covered by pollen that landed on Tommy Atkins, Van Dyke, and Palmer trees than the other 8 maternal parents. And these cultivars, Hayden, Graham, and surprisingly, Tommy Atkins, were the highest pollen donors. So most of the pollen, most of the seedlings were as a result of pollen from this strain. So it's possible to use this kind of genotyping method to select superior hybrids and with known parents to improve the selection efficiency in a mango breeding program. And I would like to acknowledge the USDA NIFA for funding this project and Dr. Edward Gilly Evans for facilitating my travel to present this work at the just concluded National Association Plant Breeders Conference in Greenville, South Carolina. And if you want to read more, we just published this paper this year in Scientia Horticulture. Thank you.
Any questions?
I have Vincent next talk and this like SNP markers to identify the parental cultivar involved in the seedling, right?
Yeah.
To me, it's like this good to tell the mother tree because the seedling next time you collect the seed about seedling, the parents, the father should be changed, right? So the mother is fixed. So you identify all the mother tree. Yeah. And the father could be contribute next year some pollinator.
Correct. So yeah, the question is whether the seedlings, if you have a fixed cultivar that you're selecting from, the pollen can come from different paternal parents, right?
Yeah.
Yeah, it can, depending on the, like if you take this year and genotype, next year, the results will be different.
Structure, yeah.
So basically, it just helps you in selecting and just taking out to the field just a manageable size of the offspring, and then not caring about the ones that don't meet the needs that you have.
Also, like you know, for squash, they said like the bee, the pollinator can pollinate your flower, you know, then like 1,500 meters are surroundings, then they can pollinate. I don't know from mango how far they can transfer the pollen. So your neighbor, like they have the mango tree, also not you don't know the parents, they also can be pollinating your.
Correct. Yeah, that can happen. And we did see that pollen can travel huge distances, even within the group. And so mango is pollinated by insects and wind sometimes, so they do travel huge distances. Yes, Ron.
So now we know which mangoes we like, right?
Yeah.
So which are the 2 parents that you will pair or cross to get a superior cultivar?
That's a good question. So I'm looking at maybe two or three major perspectives. The first one is flavor. Now, for flavor, there are cultivars that are known for the high sugar, sweetness, such as Diamond. Hayden is also really sweet. Coconut cream has a different flavor, sweet, but also tastes like a coconut. So when I'm selecting the seedlings, I'll consider the parents that have those qualities in terms of flavor. Then there is disease resistance. So Tommy Atkins, the reason why it's been so successful is because it's able to withstand, maybe tolerate to some level, some diseases in post-harvest. So that is could be one of the consideration. And also some other cultivars such as Seba, which every time I go to the group to collect fruits, I never find an anthracnose on them, even though everything else around there has an anthracnose, and it's on the leaves, but on the fruit.
Wow.
So,
What's the taste? Does it taste good? Okay, of course, yeah.
It's not good. So basically, I would target something like that. And then, of course, when I grow here we normally trim the mango tree, so it's not easy to tell the growth habit, but we have a database that shows what growth habit we have for these mangoes, and that's also something else I can consider.
And there's no barriers, right? You can cross a Asian or Indian.
Yeah, there's no barrier at all. Yeah. Even within the species, you could have some interspecific crosses. Yes, Libby.
Hi, Vincent. Thanks for your presentation. It was really nice. I was actually wondering, how did you measure the distance that the pollen would fly?
Oh, that's a good question. So I'll show you the picture. Maybe I repeat the question for the people on Zoom. The question is how we measure the distance that pollen can fly. So let's say this is the mother parent, and the pollen donor is this one. I just did a right angle triangle and did Pythagoras theorem. I just assumed pollen was traveling. Maybe the bee or whatever came this way, that way, it's just an estimation.
Okay, yeah, so actually you have also to be sure that you have only one plant of one donor.
Exactly,
Because if
yeah.
you have more donors around, you don't know where the pollen
You cannot tell, yeah. Yeah, I did find some seedlings that seemed to have more than one pollen donor. But yeah, you can't really tell. Okay. So like those ones, like I won't, maybe I'm not saying.
Thank you.
Yeah, Vincent, thank you so much. Very interesting. Yeah, just curious, is there a non-destructive methodology that you can extract DNA directly from the seed and, get all this information that we need, the resistant or the parents, before planting the seed in the soil?
Okay, oh, that's a good question. So he's asking whether there's a non-destructive method to extract DNA from the seed. Yes, I think maybe there are methods we could test. I haven't seen any in mango, but I know like when I was working in squash, we did extract DNA from part of the embryo by cutting it, extracting that, and then the remaining part, we plant it and you get a plant. But that's a good thing. I could test that in mango and see if we could save time from sowing to getting the first fruit. Yeah. Thank you.
So you mentioned that Duncan, it was very interesting.
Yeah.
And you didn't say why.
Yeah, it's because of the way so Duncan had been reported in literature to be an offspring of a mango that came from Thailand called Nam Dok Mai.
I love that one.
It's very nice.
Yeah.
Now, when I did my analysis, I found that Nam Dok Mai is not really the parent. from my genetic analysis. But the parents are Edward and Carabao. So I checked to see, you know, the more information and found a patent of Duncan, and it was patented in 1965 or around there. But Namdokmai was introduced in Florida in mid-70s, maybe 10 or more years later. So there was no way it could be the parent. So from my analysis, I saw the two parents are Edward and Carabao. In the patent, they say they know that one of the parents is Edward, the other one could be a Southeast Asian cultivar, and Carabao is indeed one.
So you also mentioned that there's a lot of synonyms? Like,
Correct. Yeah.
Like more than
Yeah. So for example, there's two cultivars from India. One is called Paheri. The other one, I mean, they could have come here in different names, or maybe the people labeled them differently. But Paheri, Bombay Green, those ones are exactly the same thing. There's the tree we have for Vallenato, and Lancetilla, exactly the same.
Are those patented?
No, the Paheri Bombay, no.
What would happen? Like if your analysis showed that two cultivars that are patent
Are exactly the
are
same.
exactly the same, or are the same as something that is not, what are the implications of that? In terms of the grower buying the plants, or the nurseries buying the plants, or?
Yeah, so it's twofold. For the growers and the people buying the plants, it means that they could use either of the names and get the same genetic material. But for the one who holds the patent, maybe there's something, happening. There could be either a registered cultivar with the same genetic information, and it's registered under a different name or something like that. So I'm not very familiar with how those patent laws apply and the intellectual property in such cases, but yeah, however, when we do this testing, even though it's showing as cells, it could be only because it's here in the group. I'll do more testing. Maybe take more Lancetillas from somewhere else, more Vallenato, and test them.
Because maybe it's not the real Lancetilla.
Yeah, it could be like it's not the real. And then also sometimes what happens so I saw this, one was Baptiste, and I saw Baptiste and Van Dyke showing up as cells, but they cannot be cells. I already know those are two different cultivars. But when I went to the group, not Baptiste, it was Turpentine. It was Turpentine and Baptiste, Turpentine and Van Dyke showing up as cells. But when I went to the group, I noticed, so Turpentine is what is used as a rootstock. Yeah. It grew some branches and developed some fruits. Yeah. And then that fruit was harvested and labeled as Baptiste or Van Dyke. So that was easy to, discard. Yeah.